Hello, and welcome to another episode of the JASN podcast series, a monthly podcast where we highlight an exciting paper from the current issue of the journal. I'm Alan Yu, Professor of Medicine at University of Kansas Medical Center and Deputy Editor at JASN for Basic Science.
Hi, my name is Jason Wu. I'm a Assistant Professor of Physiology at University of Arizona, and I am also Junior Associate Editor and Journal American Society of Nephrology.
Today, we're delighted to be joined by Doctors Shipra Agrawal and Monoj to discuss their interesting paper entitled RNA Alternative Splicing and Polyadenylation and Regulation of the Glomerular Filtration Barrier, which appears in the current issue of JASN. So to start off with, can you please introduce yourselves and tell me a little bit about your background?
Yeah, so thank you, Doctors Yu and Wu and Jason, for creating this podcast and inviting us to discuss and highlight our work. My name is Shipra Agrawal, and I'm an Associate Professor of Medicine in the Division of Nephrology and Hypertension at Stony Brook University, and I'm really excited to be here. Thank you for inviting us. So my educational and training background is in RNA biology. My PhD was in RNA virology, and my postdoctoral trainings were in mRNA polyadenylation and splicing. And then I worked as a senior scientist for a few years in the kidney disease space. And for more than a decade now, I've been committed towards both basic and translational research in the field of podocyte biology and glomerular disease. And I'm joined here with Dr. Das.
Hi, I'm Monoj, postdoctoral associated in Dr. Shipra Agrawal lab at Stony Brook. I did my PhD in cancer biology from Tezpur University, India. So my research interest how alternate splicing and polyadenylations in podocyte and glomerular genes affect slit diaphragm and kidney filtration barrier using RNA-seq and functional assay to link the RNA processes change to podocyte injury or proteinuria and to evaluate the RNA-based therapies in kidney disease.
Both Doctors Agrawal and Das. So as you just alluded to, injury to the podocyte and the glomerular filtration barrier leads to proteinuric kidney disease. And your study was aimed at elucidating the alternative splicing and polyadenylation of glomerular genes encoding the components of the filtration barrier. And this is an aspect of gene regulation that hasn't received a huge amount of attention. So can you first maybe tell the listeners what was the rationale for focusing on this aspect of epigenetic regulation of glomerular genes, and specifically, why do you think alternative splicing and polyadenylation might be important?
Yeah, so as we all know, chronic kidney disease is a major global health problem, and glomerular disease is one of its leading causes. But we really don't know much about the etiology of primary glomerular disease other than the monogenic mutations that have been identified. So also we need more podocyte-targeted therapies and better diagnostic abilities for these podocytopathies. And in order to tackle some of these issues, scientists have traditionally looked at the central dogma, which is DNA makes RNA, which makes protein, in a very simplified manner. So we've seen a rapid rise in RNA sequencing databases and data sets, including bulk RNA sequencing, single cell, single nuclear, even spatial transcriptomics, which gives us a lot of information on the quantity of the gene expression. So what we've been lacking is on the quality of the gene expression or the constitution or heterogeneity of these RNAs that are being made. So to go back to basics, when RNA is transcribed from DNA, it goes through maturation or processing steps, such as capping, where a cap is added at the 5' end, mRNA splicing, wherein the introns are spliced out, and exons are joined together to make a reading frame, and polyadenylation, wherein the 3' end of the RNA is cleaved and a long poly tail is added. So these processing steps add several layers of regulation for gene expression, which determines mRNA stability, mRNA localization, and what kind of protein isoforms will be eventually translated. And as you can imagine, a dysregulation in these fundamental processes can result in disruption of the cellular machinery. And these processes are becoming increasingly appreciated in development and pathophysiology of other diseases, such as cancer and neuromuscular diseases, but severely lacking in glomerular disease and other kidney disease space as well. So these ideas have been synthesizing in my mind for a long time due to my dual association with RNA biology and kidney disease. In fact, I had the privilege of training with Dr. Claire Moore at Tufts University, who's also a co-author on this paper, who made the Nobel Prize-winning observation of mRNA splicing event under a microscope of an adenovirus at that time, many years ago. So when I was thinking a few years ago, what kind of long-term impact I would like to make in the kidney field and glomerular disease, I asked this basic question in glomerular biology and initiated these studies, which led us to validating some of the targets we observed and which we hope will eventually lead us to a better mechanistic understanding, novel therapeutics, and advanced diagnostics in future.
That's wonderful. Thank you. So for your disease model, you chose puromycin aminonucleoside and andromycin injury. Could you tell us why they are selected and how well they model human disease?
So we selected puromycin and aminonucleoside and adramycin because they are well established and widely used model of podocyte injury that recapitulate two major clinical form of human nephrotic syndrome like minimal change disease and focal segmental glomerular sclerosis. So puromycin aminonucleoside induce podocyte injury via oxidative stress and cytoskeleton disruption, leading to food processes effacement and substantial proteinuria. On the other hand, adramycin injury lead to podocyte loss, segmental glomerular sclerosis, and tubulointestinal fibrosis, resulting in progressive proteinuria and renal function decline, consistent with the human focal segmental glomerular sclerosis. So, using both model, minimal change disease and focal segmental glomerular sclerosis, allow us to examine podocyte injury in each model and find RNA processing change shared or distinct across glomerular disease.
Terrific. Thank you for that. Yeah, so I guess the readers, and the readers will probably be surprised at how many alternatively spliced and alternatively polyadenylated messages you found. So I know the study was in large part about generating a novel data set and resource that others could use, but can you tell us whether you were able to glean some preliminary insights into how alternative splicing and polyadenylation of slit diaphragm genes might lead to functional changes in the filtration barrier?
Yeah, and that's a really important question. And you're correct in mentioning that we have created these data sets and classified these events based on their cell types, a localization within the glomeruli according to their functions as well. And we've also cross-referenced or differentially expressed data sets to identify the topmost dysregulated RNA binding proteins involved in these events. And as with all big throughput data sets, as you mentioned, it's important to validate our targets and to analyze their functional significance. And in this regard, we have found that the critical slit diaphragm members, nephrin, podosin, nephron, they use different polyase sites in their disease state on their mRNAs. So we've also focused, after meticulous selection, on two crucial candidates, TJP1 and ITM2B, which we found to be alternatively spliced. So TJP1 is a tight junction protein 1, or better known as ZO1, which has been shown to be localized to the slit diaphragm. And its podocyte-specific knockout results in proteinuria and FSGS-like symptoms. So it is encoded by a long mRNA of 28 exons, and interestingly, it can exist in two different isoforms, which either contain or exclude exon 20, which encodes for an in-frame 80 amino acid alpha domain. So while this alpha plus form is present in most epithelial cells and is important for cell-to-cell junctions, the minus form is the more dynamic form, which is present in endothelial cells, allowing flexibility in cell junctions. And these two forms also differ in how they interact with the actin cytoskeleton. So to our surprise, we found that this minus form is only found in two highly specialized epithelial cells in the body. One is podocytes, and the other is the Sertoli cells of the seminiferous tubules. And in our study, we have found a change in the ratio of these two forms in the podocytes. And we are also able to modulate these shifts with splice switching oligonucleotides with modified chemistry. Furthermore, we have new evidence that exposure of these podocytes to these oligos result in a change in their viability and also expression of key podocyte markers. Thus, we believe that alternative splicing of TJP1 alters the ratio of the dynamic to the standard form, which may have consequences to how podocytes on the slit diaphragm structures respond structurally and functionally to the outside forces during injury. And the other interesting candidate that we have identified is ITM2B, or integral membrane protein 2B, which is a newly identified member of the slit diaphragm and also part of the nephrin interactome, although in brain it has been shown to be associated with cognitive defects. So its knockout in Drosophila nephrocytes has been shown to decrease endocytosis of the tracer, which is indicator of its slit diaphragm disruption. And interestingly, it also exists in two different isoforms, short and long, which is a result of differential usage of the first exon out of the 6 exons that constitutes its open reading frame. And this results in an extra N-terminal domain on the long form, which can regulate its association with microtubules, as has been demonstrated in neurons and retinal cells, and we know are important in podocyte structure as well. So overall, because of their association with the slit diaphragm and differential interaction with actin cytoskeleton and microtubules, and the functional evidence we've shown using splice switching oligonucleotides in our ongoing studies in the lab, we believe that their alternative splicing has functional consequences in altering the podocyte and slit diaphragm structure and function. And these are some of the active areas of investigations in our lab that Monoj is pursuing.
Thanks. That's really interesting.
Yeah, thank you for those highly mechanistic findings. So in your paper, you mentioned that PPA gamma activators like pioglitazone reversed alternatively spliced and polyadenylated events. So why did you test these particular drugs, and what implications does this have for reversing damage to the slit diaphragm, which is a huge unmet need in renal medicine?
Yeah, so thank you for that question. So these studies actually stemmed from our other theme in my lab, which is to selectively target the nuclear receptor and transcription factor PPR gamma to treat proteinuric states or glomerular disease. So the activation of this receptor with FDA-approved drugs such as pioglitazone is more widely known for its role in the treatment of type 2 diabetes due to its insulin sensitization effects. And we and others have shown in the past few years that pioglitazone also has direct proteinuria reducing effects. But these classical agonists have come under massive scrutiny, as you may know, as physicians, because of their side effects. And thus, in the ongoing studies in my lab using multiple models of podocytopathies, we're moving away from this complete agonism of PPR gamma to its selective modulation with these new next generation compounds, such as GQ16, to take advantages of the beneficial effects, but ameliorate the side effects. So using these compounds to restore some of these events we observe in disease informs us of the beneficial signaling pathways downstream of these compounds, and also provide us information on these mRNA processing events that are likely pathogenic and reversible.
Yeah, that's exciting. Thank you. And so, so to sort of follow on from that, how do you think these findings in mouse models might translate to human nephrotic syndrome? And can you also comment, you've mentioned a little bit about splice modifying oligos. I'd love for you to comment on those too.
Yeah, so great question. And so that is the main goal of our studies, to make them translatable to human disease, and in this case, nephrotic syndrome. So I believe that our studies not only inform us of the fundamental mechanisms, but it identifies important targets that provides potential avenues for the development of novel RNA-based therapies such as splice switching oligonucleotides, which has been completely lacking in the glomerular disease space, but very well explored and even FDA approved for other diseases, such as spinal muscular atrophy and Duchenne muscular dystrophy as well. So the other aspect we're interested in is diagnostics. So as we all know, with focus on precision medicine and coming together of different consortiums, we're continuing to see a massive rise in the GWAS SNP database. And we've also used our database or the data set from the study to identify the SNPs from the GWAS studies in the nephrotic syndrome space, which are located in the regulatory non-coding regions of the genome. And the next step for us is to use the knowledge gained from these alternative RNA processing and the corresponding assays that can be used to functionally characterize these SNPs. And having said all this, we're also keeping in mind and accounting for the interspecies variability that exists in these events in the genome, and we're working towards that.
Thank you. So those are all exciting areas of research. So what future directions do you anticipate emanating from this work for you and for others in the field?
Yeah, so thank you for asking this question. So we believe this study is the tip of the iceberg, and studies like these will get the nephrology community and the investigators thinking about various ways to approach the understanding of the disease and perhaps look at their favorite genes or pathways in a new light or even from a different angle. And we are open to collaborations if anyone would be interested in our assays, insights, expertise, experience in this area. And as I mentioned, we're actively investigating in this area for better mechanistic understanding of the functional consequences of these events using culture, organoid, and animal models. We are also studying the use of antisense oligonucleotides or splice switching oligonucleotides to modulate and manipulate these events and in turn disease outcome. And also studying the regulatory impact of certain RNA binding proteins on these events. And as I also mentioned, we're also exploring the SNPs identified through the GWAS to study their functional consequences and hoping that the funding agencies will also appreciate these studies that need to be investigated. And we're looking forward to submitting our findings to JASN in the future.
Wonderful. Thank you so much, Dr. Agrawal and Dr. Das. That sounds like you've got a lot of work ahead of you, but really exciting stuff. Wants to congratulate you on this really beautiful paper, and thanks for joining us on this podcast from Dr. JASN and myself. Appreciate it.
Thank you so much. This was really exciting.
Thank you.
Yeah, keep sending your fantastic work to JASN.
This podcast is copyrighted by the American Society of Nephrology. All rights reserved. All content in this podcast is for informational purposes only and is not intended to be medical advice. This podcast should not be used in a medical emergency or for the diagnosis or treatment of any medical condition. Please consult your doctor or other qualified healthcare professional if you have any questions about any medical condition or before taking any drug, changing your diet, or commencing or discontinuing any course of treatment. Thank you for listening to this podcast from the American Society of Nephrology.